Okay, finally. Okay, let's talk about the lab for this week. Again, please take notes because this is the kind of information that will be on the lab exam. So we're looking at pH and we're looking at buffers. And important to remember what buffers do. Buffers regulate pH change. It's important for living organisms to have buffers because remember that changes in pH denature proteins. They cause proteins to not work correctly. And we have lots of critical proteins in the body that if they don't work correctly, we will die. So regulating pH is a really important thing in the body. And a good buffer is going to regulate pH whether it goes up or down. It wants to keep it constant. Whatever normal is for that part of the body, the buffer system serves to keep it constant. So let's say you have a beaker of water. And let's say this is water you got from your sink at home and you tested the pH with something more precise than that little color chart you had. So the color chart you all have, it's a rounded number, right? Like it's an approximation. But let's say we had an actual pH meter and we tested the pH of this water at pH 6.4, which would that's probably pretty typical for tap water. Tap water has a lot of other stuff in it, so it tests a little bit acidic. No big deal, right? It hits your stomach acid, it's going to turn to pH 2 anyway. So, which, as I've mentioned before, if you drink alkaline water, someone is selling you some snake oil because you drink that water, it goes to your stomach, it becomes pH 1 to 2. It's not going to somehow magically save you from getting cancer. So you've got this water. And remember, the cool thing about water, the reason water is an excellent solvent is because it doesn't compete with the chemistry that's going on. If you add salt to water, it becomes salt water. If you add sugar to water, it becomes sugar water. If you add acid to water, it becomes acid water. If you add base to water, it becomes base water. So if we add to this water hydrochloric acid, HCl, by the way, this is chlorine and this is hydrogen. A lot of people mistakenly think that that's a capital I. I sometimes like to write it HCl like that so you can see that it's actually an L. This is hydrochloric acid. It's a very strong acid. It's about pH 1 to 2. We're going to call it pH 2 today for our discussion purposes. If I add some acid to this water and that acid is pH 2, the pH of this water should go down instantly, okay? Because water is not a buffer. That's very important to understand. Water is not a buffer. So in the experiment this week, your entire first row on that test tray is water. Okay, you're going to have the very first cell, A1, is going to be just water and the pH indicator. Okay, in other words, what is the starting pH of your water? And then all the way across, it's just water, it's not buffer. So you're going to have water, you're going to have 1 drop of acid, which is hydrochloric acid, and you're going to have your indicator. Okay, then in this one, you're going to have water and 10 drops. And I'm going to show you that diagram in a minute that shows exactly what you're adding to each cell. Okay, then you're going to have water and 1 drop of sodium hydroxide. Okay, sodium hydroxide is a strong base. Okay, so hydrochloric acid, strong acid, very low pH. If that's added to water, we would expect the pH to go down. So pH is going to decrease almost instantly. If we have water, again, pH, I'm going to assume 6.4, and we add sodium hydroxide is a strong base. which means it has a high pH. We're going to say it's pH 12. If I add a strong base to water, we would expect that pH to increase dramatically because, again, water is not a buffer. So the water becomes whatever we add to it. If we add a base to it, it's going to become a base. So the pH is going to go up dramatically. And here you're testing that. You're adding 1 drop of sodium hydroxide, and then you're adding 10 drops of sodium hydroxide. All the way across this row with water, this is your control. This is your experimental control. The whole reason we're adding it to water is to show what would happen if it wasn't a buffer. You have to have that water data to compare to your buffers. Okay, because without the water, you don't know if buffers are doing a good job or not. Like if you just do the buffers, like, well, it changed a little bit. I don't know if that is that good. Was the buffer doing its job? The only way you know if the buffer is doing its job in controlling the pH is this: if you compare it to something not a buffer. And that's what water is in this experiment. Water is the control that you'll compare your buffers to. So the next 3 rows are really your 3 different pH values of the buffer. You have pH 4 buffer, pH 6 buffer, and pH 8 buffer. And in a perfect world, you would have had full bottles of all 3 of those beautiful buffers. But we already know that that is not the case. Okay, I'll tell you, last semester, it was the blood type kit that spilled. This semester, it's the pH kit. So we're trying to do the best we can. For most people, pH 4 is good. Everybody has enough pH 4. And honestly, I don't know how these spilled. If you've noticed, they all have that little seal on the top that is somehow popped off. And it's not only leaked out of the seal, it's leaked out of the cap. None of the caps were loose. So I don't know. I don't know how they shipped these, but there you go. Whichever one of these you're missing, one of your options is to just substitute with milk. But, you know, just substitute one of them with milk, not both, because otherwise, you know, if you're substituting both of them with milk, then that's the same thing, and that's not going to work so well. So that would be, you know, my first solution. If you want to come get buffer from me, please, you know, I am happy to put buffer out for you, or I can arrange for you to go to campus and get that. If you choose to go to campus, you'll have to go to the NS building, go into the 2nd floor, and go to a person's office, and she will give it to you. So that's an option.
This is only if it's spilled,
Let me know if that applies to you. So those next rows, you know, it's the same thing, you know, in this first one, you're just putting your buffer. So this would be your pH 4 buffer, and you're testing your starting pH because you're going to put the pH indicator. So let me just quickly, let me share, let me stop the share here for a second and show you something really important on the lab exercise. Can everyone see the frog? Okay, awesome. So there's a lot of blah, blah, blah. There's a calculation. I'm going to go through this calculation in a minute. This is incredibly important because the sodium hydroxide and the hydrochloric acid they sent you are too concentrated. You're making a dilution of those. And it's important to know how to make a dilution. So we're going to go through the math of that in a minute. That calculation is really important, and you'll need to be able to do a similar calculation on the lab exam. So we'll go through that. In addition to the materials in the pH kit, you also need some materials from your general equipment bag and your safety kit. Okay, number 1 important thing, safety goggles. Okay, the only people who might possibly see you wearing these beautiful goggles are the people in your house. Your eyes are really important. Please don't get acid in your eyes. Okay, wear the goggles. It's my number one tip for today. Wear the goggles. Okay, their eyes are important. And then you need some other materials. Please do not throw these away. You will use these throughout the whole semester. So don't throw away the materials in the general equipment bag after you use them. Rinse them out and you will be using them again. Distilled water is preferred, that meaning bottled water, but you can also use tap water this week. That's not a problem because you are going to be, I'm going to zoom in on this, you're going to be testing the pH at the beginning. In this very first well, you've just got water in your bogans, that's the pH indicator. Okay, so you have the pH indicator, you're going to be testing the pH, your starting pH. So it doesn't matter what that starting pH is. You can use tap water, it's totally fine. Again, this first well all the way down is just your water or your buffer plus the pH indicator. In other words, this is the starting pH of the water. This is the starting pH of your buffer 1, buffer 2, and buffer 3. Now, in this one, you're going to add 1 drop of the diluted acid, then 10 drops of the diluted acid, 1 drop of the diluted base, 10 drops of the diluted base. And that's the pattern all the way across. Here you have your pH 4 buffer, 1 drop of acid, 10 drops of acid, 1 drop of base, 10 drops of base. So please, this is the most important thing for you to look at when you're doing your lab. A lot of times students will email me and they'll send their picture and they'll say, my color didn't change or my color is not how I expected. It's usually because they didn't put all 3 things in the well. Okay, so everything after this first column, you should have 3 things in there. Your water or your buffer, acid or base, and your pH indicator. So really make sure you pay attention to that. Okay, and then the questions, I have reworded the questions. So the questions that you have this week are, current, they're written by me based on this activity by Carolina. You are just doing activity 1. We will not be doing activity 2. The reason we will not be doing activity 2 is because it's stupid. That is my kindergarten answer for today for why we're not doing it. I honestly, we did it in the summer, we did it last semester, and both times I was like, what is the point of this? So we're not doing it. Okay, that's why last week I just had you test the pH of household items, is because we do usually activity 1 week and activity 2 the other week. Activity 2 is no good. So we're just doing activity 1. Be sure to take a picture and that is what you're submitting. But it's important to answer all the questions because the questions are going to be on the lab exam. So let me show you how to do that calculation. And I'm going to stop sharing and then go back to sharing screen. Does anybody have any questions while I'm getting my piece of paper off the floor? Okay. I'm going to show you how to calculate, how to do the dilution this week. So it's really asking you 2 things. It's asking you how much of the acid or base do you add, and then how much water do you add to make a dilution. So let me go to here. I'm going to share my screen. Okay, so here's the story. You have a bottle of 1 molar hydrochloric acid, and you have a bottle of 1 molar sodium hydroxide. That M is telling you the strength or the concentration of that solute in the water. So it's a solution. And remember, a solution, this is from the properties of water talk. Solution is a solute plus a solvent. Okay, the solvent is water, and the solute is what's mixed in the water. So in this experiment, your solute is either hydrochloric acid or it's sodium hydroxide. What this M with an underline is telling you, the word for that is molar, and it has to do with molarity. And we talked about molarity a little bit in the properties of water lecture. Molarity is telling you the strength of that solute. Okay, so as we're moving toward talking about the cell starting next week, we're going to be talking about how materials move across cell membranes. And this term molarity is going to come up over and over and over. If I have a 5 molar sodium chloride concentration, that's 5 times greater than a 1 molar sodium chloride concentration. So it's just telling you something about the strength. And normally we learn how to calculate that strength. We're not doing that this semester because people want to come at me with flame and pitchforks when I do that. It's not something people enjoy. So at some point you'll do it in a chemistry class, and that's good enough. Okay. So this is pretty concentrated. They're asking you to do a dilution of this, and they're asking you to do a very specific dilution. You're starting with 1 molar, and you want to get it to 0.1 molar hydrochloric acid. Okay, so that's 1/10 the concentration. But you want a specific volume also. You want 12 milliliters of it. Okay, why did they tell you you needed 12 milliliters? I'm guessing that they tried to count the number of drops you're going to need for the whole tray and give you some extra. I think that's how they came up with 12. Like maybe they thought 10 isn't enough because 12 just seems weird, right? Okay, so same with the sodium hydroxide. You're going to dilute that to 0.1 molar, and you need 12 milliliters of it. Okay, if we're diluting something, we can assume we're going to be adding some water, right? We're going to add some water to dilute it, just like you would if you were drinking lemonade at home and it's too sugary or too lemony, you would be able to add some water to dilute that concentration to make it less strong. That's essentially what we're doing. The question is, how do we know how much water to add and how much acid to add to get to that concentration? The way we do that is ratios. So what they're telling you with that equation that they give you is they're saying that molarity initial and volume initial in ratio is equal to molarity final and volume final. I actually like thinking of it this way. I like thinking of it as molarity 1, volume 1 equals molarity 2, volume 2. You can think of it either way. Whatever makes more sense to you, go with that. Here's what we already know based on what we wrote up here. The initial molarity that we're starting with is what's in the bottle. That's what we're starting with. And the strength of that is 1 molar. So let's start by doing our HCl calculation. And I'm going to do that calculation for you. I'm not going to turn you loose to do that on your own, but realize you will have one of these on the test and it will have different numbers. So make sure you know how to do it. And you can just follow this pattern when you do it. So molarity 1 or molarity initial, volume initial equals molarity final, volume final. Our initial molarity is what we've got in the bottle, and that's 1 molar hydrochloric acid. What we want to calculate is this. Okay, this is going to be how much of the 1 molar HCl do we add? That's what we're going to calculate. So that's really our unknown right there. Because what we want it to be at the end is we want it to be 0.1 molar. And the volume we want is we want 12 milliliters of hydrochloric acid solution. Any questions about that setup so far before we do the math?
What did the V stand for again?
I'm sorry, Dylan, for some reason I can't hear you. Just a second, it's me. So let me, I'm so sorry, Dylan, try it again.
What did the V stand for?
The V?
Yes, on the MFVF.
Yeah, so V is volume.
Oh, okay.
And this is molarity or concentration. Great question. Anybody else with questions about the setup before we do the calculation? Okay, so for the calculation, I'm going to erase this part right here. Oops, sorry, hold on one second. My little doodad fell off my Apple Pencil. Sorry. Okay. I'm just doing that so I have room to do the math here. Okay. In case you haven't had math for a while, it's not a problem. I'm going to show you what to do, okay? So don't worry. And honestly, this is one of the last weeks we do math in this class. So please, I know math causes anxiety in some people, so please don't worry. You know, we're at week 6. This is the second time we've done math. It won't happen again. I promise. If it is, it's like doing an average. It'll be something easy. No more algebra or whatever after this. Okay, so what we want to do is we want to solve for this. Okay, to solve for that, we have to do the reverse on both sides. So over here, we're multiplying by 1 molar hydrochloric acid. So now we have to divide by 1 molar hydrochloric acid. So we're going to move this around. We're going to move that 1 molar hydrochloric acid over here. So it cancels out here. And now we have the volume of that hydrochloric acid that we have to add is going to equal 0.1 molar, 12 milliliters, 1 molar. Oops, sorry, I didn't put my hydrochloric acid here. Okay, so remember, if I divide units by the same units, this goes back to the metric conversion problems. Okay, so if I divide molarity of hydrochloric acid by molarity of hydrochloric acid, that cancels out, just like it did in the metric conversion problems. And it's a 1, so I really don't have to do it, right? So really the whole thing cancels out. So now it just becomes math. It's 0.1 times 12. So that's going to equal 1.2 milliliters of hydrochloric acid. That's how much I need to add to start to make my solution. But let's think about this. Okay, so it's saying that I need to add 1.2 milliliters of my 1 molar hydrochloric acid plus enough water to make 12 milliliters total of solution. I want 12 milliliters total. I just calculated how much of the concentrated acid to add. So this is going to be your chat question for attendance today, okay? You have to answer this question. So anyone who's kind of tuned out right now, tune in. Here we go. Here's your attendance question. How much water do you need to add? Again, no right or wrong answers here. You don't get graded on your answers. You just get graded on if you answer. Okay, so it becomes a math problem. I need 12 milliliters total. And I'm already adding 1.2 milliliters of acid. So I need to know how much water. So that would be 10.8 milliliters of water. So here's my recipe for making the solution that I need to make. And you're going to add this in your test tube. You're going to add 1.2 milliliters of hydrochloric acid plus 10.8 milliliters of water. And that's going to give you 12 milliliters of 0.1 molar hydrochloric acid solution. And that is what you add in those wells that are requiring acid. So in some of the wells, you're adding 1 drop, in some you're adding 10 drops. So remember, you're adding the diluted version, not the straight up stuff that came in the bottle. The straight up stuff that came in the bottle is too concentrated for this experiment because we just got those tiny little wells that we're working with. Any questions about that?
So for the drops, pretty much we look on the bottle and see how much like the molarity of the solution is in the bottle. And then we just calculate it like that. Like, so we put how many drops based off the solution that it says on the bottle.
No, so the directions of the lab tell you the number of drops.
Oh, okay.
Yeah, so that chart I showed you, it like some of the wells get 1 drop of it and some get 10 drops.
Okay, and then after that it says, is this molarity of solution based on how many drops it tells you?
It tells you to add either 1 drop of the diluted stuff or 10 drops of the diluted stuff. So this is just telling you how to make the dilution.
Okay.
Because the stuff you're starting with is a lot more concentrated than what they want you to use, just because it's those tiny little wells. Plus, they're just teaching you how to do a dilution. You'll be doing dilutions in micro and physio and chemistry. So it's an important skill to know. So they're just trying to teach you that too. Okay, so exact same thing for the sodium hydroxide. You're starting with 1 molar sodium hydroxide. Okay, and you need to get to 12 milliliters of 0.1 molar sodium hydroxide. So what you need to know is how much of this to add to make the solution. And we calculate that using that same exact formula. So molarity initial, volume initial equals molarity final, volume final. So we know this number. It's 1 molar sodium hydroxide. We don't know how much we add. That's what we're calculating. And we want it to be 0.1 molar sodium hydroxide. And we want 12 milliliters. So it becomes the exact same problem. We divide both sides by 1 molar. So this cancels out. I can cancel out my units and it becomes a math problem. 12 times 0.1 is going to give us 1.2 milliliters of sodium hydroxide. And I need 12 milliliters total, so plus 10.8 milliliters of water. And this is the 1 molar sodium hydroxide that we're adding, the original. So this is how we get to 12 milliliters of 0.1 molar sodium hydroxide. So you're mixing this, the directions tell you how to do this. You're mixing it in your test tube, and that has to go in your test tube rack. That becomes your stock solution for putting the drops in your wells. So yes, you're making 12 milliliters of the 0.1 molar concentration, and that's what you're adding to the cell. Now you don't add it to every cell. So if you look at that chart, it tells you where to add it. So if you divided that into, let me just kind of remind you all what you're doing. And luckily it does follow a logical pattern. Oops, I made that first row too big. Okay, this first one is just going to be, I'm just going to put a B with a circle for your pH indicator. That's the Bogen's pH indicator. Okay, that goes in every single well. In fact, if you wanted to just go through and put that in every single well, you could. So every well gets pH indicator, right? Because we want to know the pH in every single well. So that's what that is. That's the Bogen's pH indicator. I think it's actually called Bogen's universal pH indicator. Okay, in this one, we just put water. In this one, we just put pH 4 solution. In this one, we just put pH 6 solution. And this one, we just put pH 8 solution. Okay, so this whole column is just telling you really your starting pH. Now you're going to add those all the way across. So this would be water, 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 water. pH 4, pH 4, pH 4, pH 4, pH 6, all the way across, and then pH 8, all the way across. Okay, now what you add to each one is different. So this is going to be 1 drop of your diluted hydrochloric acid, and this will be 10 drops of your diluted hydrochloric acid. So how does the water change with just 1 drop of strong acid, and how does it change with 10 drops? Obviously, we would expect it to decrease more with 10 drops, right? Versus 1 drop. Okay, here we're adding 1 drop of a strong base, sodium hydroxide. Here we're adding 10 drops of sodium hydroxide. Okay, so what happens to the pH of the water with just 1 drop versus 10 drops? We would expect the pH to go up significantly more with 10 drops than with just 1 drop. But you know what? Maybe that acid and base are so strong that even with just 1 drop, that pH changes dramatically. We don't know that until we do the experiment. We can kind of predict that. So when you're making your predictions about what should happen, really think about the science behind this. Think about the logic of what should happen. Okay, and then that same thing happens all the way down. We're going to do 1 drop of acid and 10 drops of acid. All the way down. Okay, 1 drop of sodium hydroxide 10 drops of sodium hydroxide. Now with the water, remember the water is your control. So you would expect that to change a lot in pH, either up or down, depending on if we're doing base or acid. So these buffers, we would expect them to change less than the water. The question is, does one of them do a better job when it's acid involved? And does one do a better job when it's base involved? Because let's think about this. If you're starting with something in a beaker, let's go with our pH 4. I'm going to do two different scenarios here. Okay, here's my pH 4 buffer. Oops. And here's another beaker with pH 4 buffer. And in this one, I'm going to add hydrochloric acid. And in this one, I'm going to add sodium hydroxide. Okay, and again, let's assume that my hydrochloric acid is pH 2 and my sodium hydroxide is pH 12. Can this buffer regulate a strong acid and a strong base? Okay, or is it going to be better at one than the other? That's what we're kind of investigating here. So when I add something that's pH 2, we would expect both of these to be about the same, but maybe they won't. You know, maybe pH 4 really can't do a good job with this strong base because there's such a difference in pH here. But if this was pH 8, maybe it could. Maybe the pH 8 buffer can do a really good job at controlling the strong base. So that's what we're testing, is which buffer is most effective with an acid and which buffer is most effective with a base. So really think about that when you're making your predictions of what you think might happen. I find those directions in the lab to be very confusing. And I'll be honest with you, the first time I did this lab last summer, I sat down with a highlighter and a pencil, and I was trying to write out what my understanding was of what you were supposed to do, and it was not clear to me. But this to me is clear. That I like the chart because the chart is clear. And that's the conclusion I finally reached, is those directions are crazy. It's too much, but I think the chart is good. So please, you know, I really love when the lab groups are coming on Thursday during my office hour, because then if you all have questions while you're doing it, you can ask me. That's the preferred, but I get it. Not everybody can do that. So please don't hesitate to ask if you have questions when you're doing the lab this week. But that's it in a nutshell. Any questions about the lab before we go on to lecture stuff? Okay, let me just make sure that nobody else has gone into the breakout room or the waiting room. Okay. I need to talk to you a little bit about the lecture material, because it is really, really important to understand some of these concepts, because we will be building on this for the rest of the semester. So last week and this week, we are talking about the macromolecules, and we have 4 categories of what we call the macromolecules. Okay, so macro implies that they're big. Okay, these are big molecules. They're much bigger than oxygen, carbon dioxide, a lot of the molecules we have in our body. These are all organic, meaning they're a carbon-based molecule with other molecules. They're a carbon-based molecule with other atoms attached to that carbon, mostly carbon and hydrogen, oxygen, nitrogen, phosphorus, and sometimes sulfur. Those would be the top atoms that would be associated with these macromolecules. So you studied carbohydrates and lipids last week. And it's important to realize that carbohydrates are our primary energy source in the body. This is how we make ATP by cell respiration. And you're going to learn the details of how we do that this semester. So carbohydrates are incredibly important in your diet. And it's important to understand the two different kinds of carbohydrates, so simple versus complex, and the fact that the simple carbohydrates really start breaking down in your mouth. So the enzymes in your saliva start breaking down that starch into glucose almost immediately. So you get this big sugar rush. So we've all experienced that, right? Like you eat, well, I guess, the best example would be if you eat some sugary candy. Like if you eat some gummy bears or Starburst or jelly beans or, something that's just straight up sugar, you get that sugar rush right away. Same thing if you eat white rice, white bread, all the stuff that tastes good. It tastes really good because you start breaking it down to sugar in your mouth. So it tastes really good. That has a different effect on your blood glucose levels than a complex carbohydrate that doesn't get digested until it reaches your small intestine. Because remember that the small intestine, I'm just going to abbreviate it SI, it receives food a little at a time. And remember, it's really pH that's driving that, because you can't just dump all of that acidic food from the stomach into the small intestine, or you'll denature all of your digestive enzymes. That's really good when it comes to blood glucose regulation, because if you're eating complex starch, which is called amylopectin, remember it's branched in structure, it's digested in your small intestine. It can't be digested by the enzymes in your saliva. So what that means, since you're getting that food a little at a time, it means you're digesting the starch a little at a time. And this is going to be a slow, steady release of glucose from that food. And that's pretty ideal, because you need a constant supply of glucose. It's one of those factors we have to keep constant in the body. Why do we need a constant supply of glucose? Because you make and use your body weight in ATP every day. That's pretty crazy to think about. So you need a constant supply of glucose. If you have this slow, steady release, that is ideal. And you get that from eating complex carbohydrates. So brown rice, whole grain bread, whole grain pasta, the complex starch that's in fruits and vegetables, especially the really fibrous vegetables. So that is the most important thing you eat every day. And if you're not eating enough glucose, your body will do something to make glucose. It will mostly rob your organs and muscles of protein, because you can only break down so much fat a day. Fat is our long-term energy storage. So if you eat too many carbs, it goes into long-term storage, which is fat. And remember from the videos, carbohydrates have 4 kilocalories of energy per gram, whereas fats have 9 kilocalories of energy per gram. So fats are pretty energy dense. That is our form of energy storage. And it's really interesting to think that we have evolved through natural selection. Those of us who can store more fat are the ones who survived and reproduced, because we were all nomads on the plains of Africa. Okay, we didn't start farming and growing crops and staying in one place until much more recently in human history. Okay, at the beginning of the history of our species, we had to wander, constantly looking for food. And we would go days without food. So those who could store the most fat are the ones who survived and reproduced, because that's our form of energy storage, so that we don't have to be eating constantly all day, every day. So those of us who could store the most fat, our ancestors are the ones who survived and reproduced and passed those traits on to us. And now guess what? Fat is not hard to find, right? We're designed to crave it. So we crave the fat, and it's readily available. And because our ancestors were the ones who could store it really well, we store it really well. So that's one of the problems we face in our diets today, is fat is really important. And I'll go ahead and tell you straight up, there is a question on the test about the importance of fat in your diet. You have to understand how important fats are in your diet. Your normal development, your immune function, your neurological function, so many different things, production of hormones, de-stressing, all of that, fat plays a really important protective role in our bodies. So fat is really important, but again, natural selection has dictated that we are the ones who descended from those who could store more fat. Because when we were just wandering looking for fat, it was hard to find. Okay, if you're just picking berries and stuff you're finding in the wild, you're not eating a lot of fat. The only way you could get fat is if you hunted and got animal fat, or you happen to find some nuts, which was only certain times of the year. So if killing an animal was dangerous, and it took a lot of energy. If you haven't eaten for 5 days, you're not going to go chase down an animal to kill it, because that takes a lot of energy that you don't have. You need to conserve your energy. So all of these things have led us to where we are now. It's just really important to think about that, like how we got to where we are in terms of human diet, and how what we eat now is so different because of what's so easily available to us. And, in the US, fast food is more readily available. If you live in Europe, they do have really big supermarkets, but that's not where most people shop. Most people go to the bakery to get their bread. They go to the open market to get all of their produce for the week. They go to the butcher to get their meat. They don't walk down aisles and aisles and aisles of all this prepackaged stuff that's not really even food. So what we eat is really different in the United States, and also really different from what our ancestors ate. Okay, so that's carbs and fat. Those are energy sources, but fats play other roles too. But proteins and nucleic acids are something else. Proteins serve a number of different functions in our body, almost none of which are energy. Okay, so we can use protein for energy, but we don't store protein. So proteins are not stored as an energy source. If we start using protein that we've eaten as energy, we're usually taking that protein from somewhere else. We're taking it from a muscle or an organ, and that's not so healthy. Proteins are used to build other proteins. And in other words, they're broken down into amino acids. And you use those amino acids to build your muscles and your enzymes and your hair and everything that's a protein in your body, cell receptors, all those different categories of protein. So when you go through the lecture videos, you're going to see there are different types or categories of proteins. And none of those are energy sources. Most of your proteins are enzymes. So it's important to understand what enzymes do. Enzymes actually reduce the amount of energy that's needed to start a chemical reaction. And without enzymes, most chemical reactions in the body couldn't take place. Okay, really great example of something we've already talked about a couple of times now. lactase enzyme. All enzymes are proteins. So lactase enzyme is a protein. What it does is it takes glucose, which is bonded to galactose, and it breaks that bond so that they become two separate sugars. If you don't have this enzyme, there is not enough energy available in your cells to break that bond, so you don't do it. We need a separate enzyme for every single reactant that needs to either be broken apart or put together. Okay, we need enzymes for that. They lower the amount of energy required to start that reaction. Without an enzyme, there's not enough energy available. We'll talk about enzymes a lot in the energy unit, which comes up right after spring break. But that's just one category of protein that we make. We make a lot of different kinds of protein in the body. It's important to realize proteins that we eat are typically not an energy source. They're typically used to make other proteins. So when we eat that protein, it's the polymer, remember, is that polypeptide chain, which is really just a chain of amino acids. And we break that down through hydrolysis into individual amino acids. And we use those amino acids to build other proteins. Okay, so if you eat a piece of chicken, and so you eat a chicken breast for dinner, you're going to break down that chicken breast into individual amino acids, and you're going to use those amino acids to build your own proteins. So it's crazy to think all the proteins in your body are built from the parts of other organisms. So, sorry, I just got a request to scroll up, and I absolutely can. Thanks for asking that. So those amino acids that you're going to build so you ate a chicken breast, you're not now going to build a chicken breast, right? You're going to put together some enzymes, some cell receptors, some integral proteins in your plasma membrane. You might make some hemoglobin on your red blood cells. You might make some insulin, that's a protein. You might make some antibodies because you just got the COVID shot. And those are all made from amino acids from what the chicken ate. So the chicken eats a beetle, and that beetle ate some grass, and those are the amino acids that are used to build you. So you're built from amino acids from all the organisms that you've eaten and they've eaten. That's pretty crazy to think about. When you eat nucleic acids, those are only used to build other nucleic acids. So when you eat real food, you're eating a plant or an animal or some product of that, if you're eating real food. So you're eating a plant or an animal product. And in those plants and animals, you are eating the DNA and the RNA in their cells. And remember, those are chains of nucleotides. So through hydrolysis, you break that down into nucleotides. And we use those to build our DNA and RNA. So your DNA is made from the DNA nucleotides of other organisms. So unless somebody in the food chain has eaten a human, which is possible, unless somebody in the food chain of what you're eating has eaten a human, you are not building your DNA from human nucleotides. That's pretty crazy to think about. Okay, so your DNA and your RNA is made of the nucleotides going back to dinosaurs. Like you could have some T-Rex in you right now because it just all gets recycled eternally through the food chain. You're made of those nucleotides. You're made of those amino acids from other organisms. I think that's super cool. It's important to realize that DNA codes for the amino acid sequence of your proteins. This is really, really important. We're going to hit on this so many times throughout the semester, and I can guarantee there's a short answer question about this on the test. The exact wording of that is in the study guide, but I just want to really point out, really important, DNA codes for messenger RNA, and that messenger.
It doesn't look like you have an app
Oh God, so sorry. What is going on? Stop.
named Messenger. If you'd like, I can help you look for it on the App Store.
Oh my gosh. You know, really, old people shouldn't touch technology. Like, seriously. My daughter, if she saw this right now, she would be dying. She'd be just ready to like put me in the home right now. Okay, DNA codes for messenger RNA, and that codes for the amino acid sequence of proteins. I want to show you a really important, I know we have 3 minutes left, and I want to show you something really, really important, because this is kind of what we're working toward. And it's important to realize right now, at this point in time, it's important to realize if you have the wrong code on the DNA, that's going to give you the wrong code on the messenger RNA, and that's going to give you the wrong amino acid sequence on the protein, and that protein is not going to function correctly. Because remember, that protein has a specific amino acid sequence, and that's what causes the shape of the protein. So if you haven't watched the lecture videos this week, it's okay. I just want to tell you that those amino acids, so if this is an amino acid, and this is another amino acid, and this is another amino acid, what they all have in common in their structure is they all have something called an R group. And for each of our 20 amino acids, that R group is different. And when they all get in a chain, these R groups all start interacting with each other. And it's the interactions among the R groups of the amino acids in the chain that causes the specific 3-dimensional shape of the protein. A protein must have the right shape to function correctly. So wrong shape equals wrong function. It won't function correctly. If you have even one amino acid in the chain that's different, that's the incorrect amino acid, it's going to have a different R group. It's not going to interact the same with everybody else in the chain, and it's going to give you the wrong shape. How do we get the wrong amino acid? We get the wrong amino acid when we have the wrong code on the DNA. The wrong code on the DNA is going to give you the wrong code on the messenger RNA, and that's going to give you the wrong amino acid sequence of the protein. So even just having one base that's wrong can give you the wrong shape of the protein. We're going to talk a lot about genetic disorders in this class, and the basis for every single inherited human genetic disorder is it's the wrong base on the DNA gives you the wrong base on the messenger RNA, which gives you the wrong amino acid in the protein. The protein has the wrong shape, and therefore it doesn't function correctly. And, you know, the severity of that depends on what protein we're talking about. Like, you know, if we're talking about a protein that gives you nice, thick, lush hair, so then you have thin, stringy hair like me, big deal. You're not going to die. You just have to use more hair product, right? But what if it's a key enzyme for survival and you don't make that correctly? that's going to be fatal. So the severity of the disorder depends on what protein we're talking about. And I know I'm a little over, but I just want to give you one example of how critical this is. You have 2 billion base pairs on your DNA. Okay, so that's 4 billion nucleotides. Nucleotides are the building blocks of DNA, RNA, ATP. There is a disorder called Tay Sachs that you might or might not have heard of before. In Tay Sachs, one base pair, in other words, 2 nucleotides out of 4 billion is incorrect. And that leads to, so this is on the DNA, and that leads to the wrong nucleotides on the messenger RNA, and that leads to the wrong amino acid in that protein. Why is this a big deal? It's a big deal because of what this protein is. It's a lipase enzyme. Okay, again, enzymes end in ASE and they tell you what they do. This one digests lipids, fats, okay, that would otherwise accumulate in the brain. If you don't make that lipase enzyme correctly, what starts happening is fats start building up around the brain and start strangulating and killing the brain cells. These babies are born totally normal. And usually by about the first birthday is when the pediatrician notices that that baby is missing some developmental milestones. By age 2, those babies have gone deaf, blind, they're having up to 100 seizures a day, and they die a horrible death by age 3 or 4. All because one base pair on the DNA is wrong. And it gives them one amino acid in that protein chain that's wrong. And that protein doesn't fold correctly. We're going to talk a lot about the technology today that will hopefully in the future be able to fix things like that. But for right now, we can't do that. We can't fix it. So that baby has the wrong code in every cell of its body. And it's because the wrong base on the DNA gives you the wrong base on the messenger RNA, which gives you the wrong amino acid in the protein, and that protein has the wrong shape. So this is, if I could pick one concept for the whole semester for you to leave this class really understanding, it's this one. And we're going to touch on this over and over. Those variants in the COVID virus right now, the coronavirus variants that we're seeing right now, there's a change in their genome that's causing a different amino acid in the protein. And in some cases, it's the spike protein on the surface of that virus. So that virus has an envelope with spike proteins. And the way we stop this virus is we have antibodies that we develop against that specific shape of that spike protein. And so if there's a change on the DNA, well, in this case, these are RNA viruses, so it would be a change in the RNA of their genome. And that one little subtle change in their RNA, say that there's a little slight mutation right here, and now this has a slightly different shape. Now our antibodies don't work anymore against that. By the way, the vaccine that we're using right now, the Pfizer and the Moderna, they're giving you the RNA that codes for that spike protein, and you're making those spike proteins right now in your cells. And we'll talk more about that as the semester goes on. So this concept of DNA, RNA, protein, we're going to hit on this in so many different ways for the whole rest of the semester. So if you don't understand it after watching the lecture videos, okay, this is a very condensed version of it, right? If you don't understand it after watching the lecture videos, I beg you to please come to office hours this week. Again, my office hours, if you don't know yet, Wednesdays 9:30 to 11 and 3 to 4, Thursdays 9:30 to 11 and 4 to 5. Or you can make another arrangement with me, but let's talk about it if you're not understanding it, because we're going to talk about it in so many ways. And I know that for most of you, that material is going to be really interesting and you'll understand it so much more if you get this concept early on. Okay, I know I've gone over, I'm so sorry. If you have questions, feel free to stick around. I'll stick around as long as you need me to answer any questions that you have, but otherwise, everyone else have an awesome day, and I'll hopefully see some of you on Thursday.
Thank you.
Thank you.
Thank you.
Thank you. Have an awesome day.
Bye, thank
You-
you.
You
Bye.
too.
Thanks.
I just wanted to say that for when we had to put a number, like the answer, my computer was lagging a lot. And like, I don't know if you even got my reply to it.
Yeah, I finally did a few minutes ago. I saw it pop
Okay,
up.
okay, yeah, because I don't know, my Zoom was acting really weird.
Okay, no problem.
Thank you.
Uh-huh.
Are we able to work on the cell structure and function lecture, or do we wait?
Oh no, you definitely want to wait. So okay, so you've already taken the exam and you want to start working ahead for next week?
Yeah.
Yeah, okay, that one you can do, just don't go to cell membranes. I'm actually recording today and tomorrow. I'm going to re-record that. So cell structure and function, those are brand new. So yes, you can watch those. Thank you
Okay.
for asking that.
Thank you.
You're welcome. Does anybody else have questions?